I'd like to invite the speakers to rejoin us for our discussion panel, which will be moderated by Dr. Jeremy Fry of ProImmune, our sales director. Dr. Jeremy Fry gained his Doctor of Philosophy under the supervision of Professor Catherine Wood from the University of Oxford, developing gene therapy strategies to induce immunological tolerance in transplant recipients. Jeremy joined ProImmune in 2001 to generate a new class of MHC multimer staining reagents. And for the past 20 plus years as ProImmune's director of sales, he has led the sales team in growing business, focusing on developing and implementing innovative technologies that radically improve our understanding of immune responses. Jeremy?
Great. Thank you, Emily. Hopefully all of our speakers are able to join us. Thank you, Yoonjoo and Jennifer as well, and Narasimha for your talk there as well. Thank you so much for our speakers to join us. Peng, can you hear us okay? And Narasimha, can you hear us?
Yes.
Yeah, perfect. Okay, you're here too. Okay, well, we will continue our discussion with those who are here, so that's great. Actually, my first question is, obviously we've had a really great range of different talks and a whole range of different topics. So it's going to be difficult to have like one thing that obviously draws everything together. But there are some topics which I think are, and part of the reason for these sorts of meetings is to enable participants to kind of experience maybe areas of research which you're not necessarily thinking about every day. And we feel that's of great value because it really feeds into maybe your thinking of your own studies and how you address specific challenges. So one question I have maybe for Yoonjoo, just to kind of kick things off, is how you see the field of in silico prediction sort of evolving over the next 5 years. Obviously, there's been rapid advances in AI in general terms recently anyway. But what do you think also are going to be those biggest challenges in inverted commas predicting immune responses? Because we understand there was great promise and also great challenges in regards to predicting immune responses. And my particular question is in the context of MHC class 2, where it's all very well that we can, we have good input data in relation to HLA-DR. Jennifer was talking about DR4 and the associations with autoimmune diseases, and we know that's obviously many different MHC class 2. But specifically DP and DQ, where you have more complex chain pairings of the alpha and beta chain, the input data for peptide binding is quite a challenge. So that's kind of one part of the question. And also, how do you think that sort of feeds into not just thinking about MHC peptide binding, but actually more importantly, probably downstream functional T-cell responses? Because one thing does predict binding, but actually what it actually means and whether there's a functional T-cell response is a bit more of a challenge. So yeah, we just value your perspective on that. And then again, if any of our other speakers can kind of then respond to that in the context of your own work, that would be wonderful.
Yeah, well. So I have used the in silico tools quite a lot, and I myself developed the tools as well from utilizing the publicly available data. So due to the advancement in the SCM-SMS the technology, now we have really big data for peptide MHC interactions, but the problem is, although there are a lot of data, compared to the past, it's still quite small. Thinking about the complexity of the immune, the combinations. So the weakness, the strengths and the weakness of the AI technologies is the size of data. So the maximum of the data science is garbage in, garbage out. So you need really great data, to produce really good results. But unfortunately, the publicly available data, they are generally, they are produced by all different people, and as that they are not really clean data. So fortunately, the prediction, the models, they're based on the AI technologies, is great at predicting non-epitopes, but the predicting immunogenic epitope is really difficult. You can, as you pointed out, it's not just the antigen presentation. So the unit, the T cell receptors play some roles in here, and for the moment, we don't have much data on TCRs, the PMHC interactions. So for the moment, And I think in the forthcoming the couple of years, it may be impossible, to predict precisely predict the immunogenic peptide. But for the purpose of the immunizations, we need to identify the peptide is non-immunogenic. That is quite correct at the moment. So for the moment, the people are investing lots of effort, to make the TCR, the PMHC interactions, but unfortunately, it's slightly better than random. So it's better to have it, but it's not really reliable at the moment. Yeah.
Yeah. Jennifer, what about you and your context for? I don't know whether you've ever had to use any sort of prediction tools, or what your opinion is of them in your experience, or how you view them at the stage of research that you're at.
So yeah, I've not had much experience of them at all. I mean, I did wonder if Yoonjoo's, platform that you've made is. I mean, you said it's not great for looking for immunogenicity, but I noted you did say you were looking for sort of neoepitopes in cancer settings. And I wonder, has it been applied, or is there evidence that it might be able to be applied to look for autoantigens at all?
Well, I mean, the people do use. I mean, anyways, it's better than random, so it's always good to have it. I mean, it's well, just like you showed, it's like the finding a needle from the haystack. So, if you have no guidance, then it's quite difficult. But the computational methods at least gives such guidance, and as the prediction accuracy, it's slightly better than random. So I think there's the really utility in terms of the of the computational method, but the people do use it. But what I'm just saying is that it's not as accurate as you expect. I mean, compared to the AI technologies is in other fields.
And I guess
another thing.
Oh, sorry.
No, you go ahead.
About functional responses, and it's the context of the presentation of the antigen. It's so important, but and that one, I guess, is more difficult to capture in these in silico environments.
Yeah, 100%. And actually, following on from that, you're talking about the context of presentation. Part of that story is actually the combination of MHC molecules that are being presented on the surface. So if you have a situation where you identify using an in silico prediction tool, which is maybe, let's say it's quite accurate, it identifies the particular peptide combined with a particular MHC molecule, what it doesn't necessarily think about in the way that it's designed is what that combination of HLA alleles is going to be on the surface of that particular cell. And if you then have that competitive situation where peptide will bind to one MHC type over another, it's all going to depend on the combination of alleles that are actually present on that patient, which is why we often see, and obviously there were questions earlier about how many patients should you have in these sorts of immunogenicity assessments, but actually maybe what specific HLA type coverage do you need to have? It's a great question, but actually I think it's probably too simplistic to think of it like that, because ultimately, even if you ensure that you have the same HLA type of donors covered, you won't necessarily always see the same outcome, because ultimately it's different combinations of MHC that are present. So you've got different presentations occurring. You've got maybe other factors as well, which we're not even aware of yet, which can contribute to how peptides are presented on MHC. So there's a lot of work that we haven't really thought about, or we have thought about, but we don't really know how to address it in these kinds of prediction tools. So yeah, it's quite interesting. Narasimha, just because we can't see you doesn't mean to say you're not there. What's your view, particularly in the context of impurities? Because obviously what the FDA guidance talks about, using in silico tools to predict potential binding of these sorts of generic peptide impurities to specific MHC types. What's your kind of thought about that?
Yes, Jeremy. In my view, in silico tool prediction, I feel that it's in a very basic stage, but still what I believe is because of the data available, we can do the assessments of these impurities covering the global population. At least I believe 95% should be able to cover in this in silico tool. But still, again, this in silico tool is a prediction model. So agency also recommends further investigation using in vitro assays. So in silico prediction should be followed by in vitro testing again to confirm the results. And
Great.
one point to add is I think current in silico tools are having 2D interactions. Maybe in clinically it's all 3D interactions. So I believe that a lot of improvements or betterments could be adding up into this in silico tool developments in future, and in combination with AI again.
Thank you. Peng, I know
Thank you.
I think thank you very much, Ramata. Peng, you've mentioned, I think you've joined us as well. I just wanted to invite you into the panel discussion. Hopefully you can hear us okay. So just to ask you as well at Eastside, have you also had the opportunity, or what's your opinion on the application of these sorts of prediction tools in the selection of specific candidates that you're then taking forward? Is that something that you can make a generalized comment on?
Sorry, I just come back now. Could you repeat the question again? Sorry for
Yeah,
that.
of course. No problem at all. Yeah, I was just wondering if you had. We were just discussing kind of in silico prediction,
Yes.
and kind of what the future holds, and maybe some of the challenges associated with that. I'm just wondering from your perspective, rather than thinking about either in the sort of sequence realm, or even
Yes.
in whole proteins, what's your own experience of applying in silico tools to selection of specific candidates in your programs?
Okay. Thank you. Thank you. Yeah. Actually, before the selection, before the assessment, we had the candidate 11 candidate compound. All of the compounds show very good in vivo anti-tumor efficacy. However, some compound show weaker anti-tumor activity, but with a very high high concentration. It means that the efficacy and the concentration it show inverse correlation with each other. So that's why we have to think about what happened for our drug. So we like to assessment the first we think the maybe our protein induced ADA anti-drug antibody, which can reduce the efficacy. So but before that we as Admiral mentioned, we have to cut we want to save time and cut the cost. The best approach for us to by assessment our drug by using in silico and GCT assay. Unfortunately, we conduct in silico, to evaluate the immunogenicity, to rank our compound by using the T-cell score. However, the drug, shouldn't sometimes not good data, or it means not correlated with the T-cell score. So actually, another choice for us to evaluate it is to use the DCT cell assay. It means as experts or professor mentioned that it can directly evaluate the T cell response to the DC. Yeah. So finally, but also we use the PD biophysical and other inhibition and affinity assay and other stability, thermal stability, and yeah, we conduct all the package, for the protein development. So finally, we selected the lead candidate. Not only we ranked the score, including T cell score, including in silico, including DCT cell and other items. So finally, we select our lead candidate compound. So now our lead candidate compound show very good data, not only extend the treatment schedule, because before the optimization, I forgot one key point is that after the DCT cell assessment, we conduct the optimization by remove the hot spot for immunogenicity and optimize our drug. by using another tool. It means in silico it is not enough for our drug, but also it is a good tool for us to, finally we conduct the optimization. So currently our lead compound is a little bit different from the from our pre-optimized compound. Yeah, it shows very good in vivo efficacy, but also show very low immunogenicity. Yes, because before that, our drug just treat the mice with twice a week. The mice showed very severe body condition change, but after we optimized the drug, it reduced the immunogenicity. So we now can treat mice with longer schedule for 2 or 3 weeks. Yeah, it should go the data. Yes. Yeah. That's my experience.
No. Yeah. No, thank you. That's a really helpful kind of context. So I mean, we've got a couple of minutes left of our discussion. I thought it might be good just to sort of understand. Jennifer, I could come back to you, and again, if the other speakers maybe just think of a short sort of aspect of this affects you, is Jennifer, you're talking about citrullination and obviously these post-translational modifications. This is a key aspect in your research in RA, and there are many different types of post-translational modifications which proteins, therapeutic proteins, generic peptides can potentially undergo following exposure, whether these are native proteins or whether these are therapeutics. Are there any, how do we kind of address that in the context of immunogenicity, where we've got a test product, but we don't necessarily know exactly the modifications that it's undergoing in vivo? Any thoughts? Big question for a minute's answer.
Yeah, I mean, in terms of our research, I guess, trying to focus on the process by which those post-translational modified proteins are generated in an immunological context. I think for us, that's where we're focusing our attention. So it's not necessarily the epitope itself, the citrullinated epitope. It's the process by which it's generated and the context in which it's presented. And I think for us, it's looking at unraveling those processes is where we're going to focus going forward.
Great, thank you. Anybody else want to kind of comment on PTMs and how we can think about that in the context of unwanted immune responses?
I mean, I guess class 2 peptides as well, they have these sort of flanking regions as well, don't they, that are quite important. And I don't know, is that something that's built into these in silico tools? Is that something that they think about when looking at peptide binding to class 2?
Well, I mean, the in silico tools, especially the MHS class 2, the one questions that in silico tools is the finding the core regions, and at the moment is the in silico tools are, I mean, it just fails, to find the core regions. So I think the key answer can be answered by structural modeling rather than the sequence-based the deep learning models. But recently the AlphaFold and the Rosetta Fold, the team is at Washington University. They developed the peptide MHC the binding prediction method, but the accuracy is not. I mean, it's comparable to the sequence-based method, but it didn't overcome the difficulties. I mean, it's like the finding the core regions. But I think the structure-based method is the key. I mean, structure-based method plus a deep learning model, that can be the key, to improve the flanking regions and as the finding the core region. But yeah, I mean, that's the way to go. But I mean, yeah, at the moment, nobody can do it, unfortunately.
Right. Well, there's a great positive note to end on. So thank you, thank you so much. I mean, there's a lot to learn, and that means we're going to be in a job for a long time to go, I think. Thank you again to all of our speakers participating, Narasimha as well. Thank you for joining us, Yoonjoo, Peng, and Jennifer. I'm going to hand over to Emily now, and she's going to take us into our next session. So yeah, thank you to all of our speakers.
Thank you.
Thank you very much.
Thank you.
Bye-bye.
Thank you.
Thank you to the speakers, and thank you, Jeremy.